introduction, and we just finished talking about proteins that are everywhere in the body, and I think we're moving up into the brain, particularly in the central nervous system. We will have 3 speakers, 2 from Lithuania and 1 from Latvia. The first speaker is Jokūbas Žiburkus from the University of Houston, talking about neurocapital abroad, strengthening the bridges with the Lithuanian biomedical science industry. And I'm also surprised it is also me. So I will start by first telling you the outline of this talk, and I have to watch my own time, so I'll try to be good. It's very simple. It will consist of the neurocapital. I will describe to you mostly my work and summarize very quickly the work of other neuroscientists, particularly in the United States. We'll talk about the examples of existing collaboration, although we didn't have much time, so I had to take those slides out. I will just mention them briefly. And finally, the organizers of this conference, to whom I'm very grateful for having me here, asked me to challenge myself and think about how expatriate scientists like myself and others, especially from younger generation, could be returning back. So for that, I challenged myself, and I came up with this vision of Lithuanian biomedical sciences super center, using the existing infrastructures and new concepts that are existing elsewhere. So the next few slides is going to be a summary of the neuroscientists that I reached out with the help of the Lithuanian Neuroscience Association. This is myself. The information is listed here. My interest is neuroscience, specialty is neurophysiology. You can see the list of the latest publications. And the stories that I'm interested in is epilepsy and Alzheimer's disease and its relationship to epilepsy. Now, at the bottom of each slide, there will be also contact information. And so if you're interested in any one of these scientists but don't have enough time to jot down information, I have it available, and you're welcome to ask me about it later. Jokūbas Žiburkus is another person I'd like to highlight. He's an MRI specialist. He works now as a senior engineer at the Intellisys Corporation in San Diego with very high-profile publications and high index of citations. Translational aspects of his MRI, functional MRI work, is on the mechanisms of multiple sclerosis as well as visual attention. Skirmantas Janušonis, who is in UC Santa Barbara, is a neuroscientist, neuropsychologist. His interests are mainly in the developmental and dysfunctions of the serotonin system and autism spectrum disorders and potentially peripheral markers of brain serotonin abnormalities. Finally, I'd like to mention one more colleague who's not a neuroscientist, but we have a pretty strong representation here from the University of Houston, the Vice Chancellor from the University of Houston giving one of the plenary talks next door, one of the main talks in this session. Oderson is an extremely talented scientist. He's more of a physicist. He's at UH. He's a medical physicist who's into sensors, has produced a very successful company, Endomagnetics, working on early cancer detection of breast cancer, and interested in magnetic hyperthermia, intraluminal magnetic sensors, and is working with nanoparticles. So I just want to let everybody know that besides neuroscientists, we have a huge capital, we have huge scientific capital abroad. Now, let's look at some of the work examples that come from my laboratory. The main subjects of study are epilepsy. In particular, we're now concentrating on severe myoclonic epilepsy in infancy, or Dravet syndrome, which is described by symptoms of febrile seizures. This is happening in the first year of life. Those febrile or afebrile seizures sometimes turn into severe epilepsy with status epilepticus. 30% of patients are attractable to the current medications or cocktails of those medications, with 20% mortality in patients, as well as in transgenic animal models. The patients will have developmental abnormalities and other dysfunctions, including autism spectrum disorders, learning disabilities, and ataxia. So this model can be very well recreated, and also in humans you can see that the mutations in the voltage-gated sodium channel, there's many sites where these mutations will produce Dravet syndrome, can account for 80% of all the Dravet syndrome in humans. So to study the pathophysiology of the syndrome, we use a transgenic mouse model. It's a knock-in. We've imported it from Riken, Japan, which SCN1A gene mutation causes a dysfunction of voltage-gated sodium channels. The two main interests in the epilepsy field I have is basic mechanisms of synaptic dysfunctions and febrile seizures, of course, with one thing in mind of how we can best control febrile seizures. The second line of research that I have in my laboratory is Alzheimer's disease, which is the most common form of dementia, with early signs such as disorientation and loss of memory. And what's remarkable that lately it is thought that up to 20% of patients, Alzheimer's patients, are experiencing seizures. Seizures may not be equal to seizures. They do not necessarily have to have a motor component. They sometimes can be silent seizures with mumbling or staring or disorientation. And so this is a very interesting topic of study. It's an emerging topic of study. To study this disease, we use the transgenic mouse model that has a human form of amyloid precursor protein, or HAPP, that forms the beta amyloid plaques, one of the hallmarks of the Alzheimer's disease. The two major interests there are inhibitory neuron dysfunctions, what, where, and how, what stage of the pathology these dysfunctions occur, and what circuits and how they happen. Again, the idea here is to control seizures and hyperexcitability in Alzheimer's disease through managing this inhibitory dysfunctions. Levels of study that we use in the laboratory range from cellular to whole animal. In cellular, we use microscopy. We produce brain slices. This is an example of hippocampus, a structure that's responsible for learning and memory, as well as is highly susceptible to seizures and epilepsy. We can target individual cells with electrophysiological recordings. We can look at the networks. For example, this is a network of Alzheimer's mouse that is studded with these white dots, which are actually beta amyloid plaques. So an example of what we can do in these models is we can target inhibitory cells that are nearby these plaques, and we've discovered the inhibitory cells in the APP mice fail to produce action potentials. This has been now funded by Alzheimer's Association. The task here is to determine what are the causes of this failure to fire action potentials. Can we restore the firing in the inhibitory cells, therefore alleviate some of the symptoms in Alzheimer's disease? The other level of study, we target neurons, individual neurons and neural circuits. We use fast functional imaging. We use voltage-sensitive dye imaging, which are small molecules that get embedded in the plasma membrane. The fluctuations in the plasma membrane potential will then cause the fluctuations in the reflective properties of these dyes, giving us a signal. So the latest publication that we have using this technology, voltage-sensitive dyes in vitro, together with electrophysiology, is in Journal of Neurophysiology. We're now pushing the envelope with some of the imaging studies and are delving into the sodium and potassium sensitive dyes. Those are dyes that are sensitive to ions. In this case, it's a sodium dye-filled cell, and we can see actually measure fluxes of sodium in dendrites, in processes like axons as well. And we can do it in single cells, or we can do it in clumps of cells, monitoring rises in heterogeneous cell populations of this ionic sodium flux. So now, the other thing that we do is using this mouse model, we produce a really nice hypothermia-induced or febrile seizure-like events in the dish. This is remarkable because we can take a slice of the brain, we can pluck it underneath the microscope, like I showed you earlier, we can raise the heat, and we could produce these seizure-like events, very much like what happens in vivo. So this is a superb model for drug testing and even drug target discovery. But that's not enough. We really want to know what's going on in vivo in these animals. So we build a heat chamber, and using this mutation, mouse mutation, we can very reliably induce seizures in these animals. It would be full-blown generalized seizures. And now we can test the same drugs that we tested primarily in vitro, in vivo, and we're doing so right now. So this is an example of seizure behavior. These are just pictures. But remarkably, in heterozygous animals with a sodium mutation that causes epilepsy, we'll have 100% incidence of seizures. But then when we use an adenosine compound that we're currently exploring, we would get a massive reduction in the seizure threshold and duration and so on. And finally, we have a collaboration on EEG recordings in the brain, closing this loop, essentially from single cell to networks of neurons to behavior and activity of the whole brain, closing the loop and actually knowing what is the activity of the brain and how we can change it with these different compounds. So the second part of the talk is going to quickly go through some of the things that I think would make Lithuania even more attractive to the expatriates. And one thing that came to mind immediately is joint postdoctoral exchange programs. The reason why is that, based on my experience in the United States, it's very easy to acquire a postdoctoral position. It is much harder to acquire a faculty position that can take 2 or 3 years. Therefore, the talented postdocs from Lithuania would be in great position to draw on expertise abroad and then be brought back into the existing infrastructure with some labs, for example, in the valleys. New grant mechanisms for new collaborations with R&D sectors in Lithuania. is important, full and part-time competitively compensated positions to neuroscientists or scientists in general already working abroad and are successful. Additionally, I'd like to mention that many young researchers, including myself, dedicate substantial amount of time in grant writing and reviews. Therefore, from the very beginning, year 2 or 3, we're already reviewing some of the top-notch research in the journals and in the grant reviews. So the specific required knowledge could be very valuable for Lithuania, and we're happy to share our knowledge in that field. That way, ideally, grants by the same collaborating research teams could then be sought in Europe and in the US, basically using European and the US funds. So again, the life sciences meeting that we're here directly strengthens and builds new relationships between Lithuanian scientists, other scientists, and Lithuanian scientists abroad. And I will finish with this vision of Lithuanian Sciences Supercenter. So it was much like a dreamlike vision. And what I propose is that it would be wonderful if we could have the elements of Genelia Farms or Howard Hughes Medical Institute, elements of Cold Spring Harbor laboratories where the professors on a rotational basis, 2 or 3 year basis, have the laboratories that are well stocked and equipped and they can touch on them for short-term collaborations and spend 3 to 6 months there, and already existing infrastructure of the Valley's and biotechnological companies. So, to summarize, Genelia Farms, Howard Hughes Medical Institute's public-private institution where you have permanent scientists with narrowly limited funding for 5 to 10 year contract, no teaching. If we really want to accomplish a lot in science, we should really think about how to alleviate teaching loads on some of the Lithuanian scientists here and having them concentrate on the research. And so the narrowly unlimited funding, of course, is on a competitive basis, and this would be dedicated to the scientists that want to come permanently to Lithuania for 5 to 10 years, or Lithuanian scientists that are already accomplished and want to take advantage of this system. Cold Spring Harbor Laboratories, of course, it's a wonderful institution, but they also have wonderful summer and winter programs. 3 to 6 month visiting positions with high-tech equipped labs on the 2 or 3 year renewable contracts. Lithuanian biotech valleys would be the third most important element in this. New academic, industry, and partnership relationships would be necessary and could be built. Public and private cooperation, business incubators, and also increased visibility to venture capitalism. Therefore, the funding here at the bottom is extremely important, and I think that funding should come from more than 2 parties, 2 or 3 party funding, public, private, government, foundations, and so on. And finally, to encourage a lot of Lithuanian scientists to come back, we must change the dual citizenship law. Dual citizenship will encourage a lot of scientists to come back, especially the ones that do not have Lithuanian citizenship but can have it. There are also a pool of people we're not exploring right now that could potentially come back to Lithuania. I'm very sorry if I went over a minute. It's difficult for me to keep track of myself. These are the people that contributed to the projects. I have a number of ongoing collaborations, several funded collaborations, and also several funded projects in the lab. And I thank you for your attention, and I think I will not take any questions to spare time for other speakers. Thank you.